Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. In this lecture, I will be going over a chemistry of nucleotides. So basically, I will be going over definitions of nucleotides, nucleosides, and then different bases that are found in nucleic acids, that is DNA and RNA. Let's get into the details. Now, nucleotides, these are precursors for nucleic acids. So, we have two types of nucleic acid, that is ribonucleic acid, which is RNA, and deoxyribonucleic acid, that is DNA. Now, the difference between RNA and DNA, it lies in the type of sugar that is found in each one of them, and also some of the bases that are found in each of these. Now, what are the other functions of these nucleotides other than being part of nucleic acids, that is DNA and RNA? So, other than being part of nucleic acids, so nucleotides, they can participate in energy metabolism in the form of UDP glucose, UDP glucuronic acid. So, it can form, they can participate in detoxification process, they can participate in a variety of biological functions and synthetic functions in our body. So, they are also participating in regulation of enzyme activity like cyclic AMP, cyclic GMP molecules, G protein coupled receptors, which basically these are nucleotides associated molecules. And nucleotide sugars, they are participating in biosynthetic reaction like glycogen synthesis, UDP glucose, which is participating in synthesis of glycogen, the UDP glucuronic acid, UDP glucuronate, it participates in conjugation of bilirubin or detoxification of drugs. So, like this, there are variety of other functions to nucleotides in our cells. Now, nucleotides nucleotide is a combination of nitrogenous base, pentose sugar, and phosphate. That's what is a nucleotide. So, that means to make nucleotide, you need a base, which is a nitrogenous base, and then you need a pentose sugar. Usually the pentose sugar, it is a ribose, or it can be deoxyribose, depending on whether it is DNA or RNA that we are getting into, and it will be having phosphate. So, this is what is a nucleotide. Okay. So, if phosphate is not present, we call that particular molecule as nucleoside. So, nucleoside. So, nucleoside is nothing but it is a base plus sugar, pentose sugar. So, if you add phosphate along with it, so nucleoside will become nucleotide. So, that's the difference between a nucleoside and a nucleotide molecule. Now, let's look at nitrogenous bases. So, we have two kinds of nitrogenous bases. One is purine base and the other is pyrimidine. Now, the purine ring, it has 9 atoms in a purine ring, whereas a pyrimidine ring has got 6 atoms. So, that's what is shown in the figure here. Look at purine ring here, where there are 9 atoms present in a purine ring. And these atoms are numbered in anti-clockwise manner. So, because it starts from N1 here, that is nitrogen 1, carbon 2, nitrogen 3, carbon 4, carbon 5, and then carbon 6, nitrogen 7, carbon 8, and nitrogen 9. So, there are 9 atoms in a purine ring. So, they are numbered in anti-clockwise manner. Whereas pyrimidine ring, it has got 6 atoms and they are numbered in a clockwise manner. So, this is little difference here between purines and pyrimidines. So, what are the purines and pyrimidines that participate in nucleic acid formation? So, the purines are, we have adenine, which is generally abbreviated as A, and then guanine, which is abbreviated as G. Coming to pyrimidines, we have thymine, which is abbreviated as T, cytosine as C, and uracil as U. Okay, so these are, so that means there are 3 pyrimidines and 2 purines. Okay, now these 3 pyrimidines and 2 purines, these are the ones that participate in DNA and RNA formation. We are going to look into what all the purines and pyrimidines that makes DNA, what all the purines and pyrimidines that make RNA later. Now, nucleosides, as I told you already, so the corresponding nucleosides is what? Nucleoside is a base plus sugar is a nucleoside. So what is the base plus sugar in terms of purines and pyrimidines? So adenine, if it is added with the ribose, it becomes adenosine. Guanine added with the ribose, it becomes a guanosine. Okay, that's how we need to call it as. And cytosine, if it is added with ribose sugar, it becomes cytidine. That's how we name it. See, cytosine becomes cytidine, whereas purines, adenine, it becomes adenosine. Guanine becomes guanosine. So SINE. INE taken out and SINE is added towards the last part of this word. And whereas it is kind of reverse in pyrimidines, cytosine becomes cytidine, uracil become uridine, and thymine become thymidine. Okay, DINE is added here. So this is a little bit difference in terms of calling nucleosides compared to free bases between purines and pyrimidines. Okay, adenine, adenosine, guanine, guanosine, cytosine becoming cytidine, then uracil become uridine, thymine become thymidine. Okay, so that's how the nucleosides will make. What about nucleotides? Nucleotides, these are phosphorylated form of nucleosides. So that's the difference between base, nucleoside and nucleotide molecules. So this table is showing you the base, nucleoside and nucleotide here. For adenine, it is adenosine is a nucleoside and adenosine monophosphate is a nucleotide. Similarly, guanine, guanosine, guanosine monophosphate, cytosine, cytidine monophosphate, uracil, uridine monophosphate, thymine, thymidine and thymidine monophosphate. So these are the corresponding nucleosides and nucleotides for individual bases that you must be aware of. Now, nucleic acids like DNA and RNA, these are polymers of nucleotide units. That means more and more nucleotides joined with one another to make a nucleic acid. So it means adenosine monophosphate, guanosine monophosphate, then cytidine monophosphate, uridine monophosphate, things like this joining with one another, giving rise to polynucleotides or polymers of nucleotide unit. Now the nitrogen bases attached with a pentose sugar predominantly in anti-conformers, which is most common one. Although the nitrogenous base attached to pentose sugar in both the conform, like syn conformer, anti-conformer, SYN syn, that is shown here, syn conformer and the anti-conformer. So this is how a base can be attached to sugar, syn and anti-conformer, but anti-conformer is more common than syn conformer. Remember that. Now nucleotides are polyanions due to phosphoryl group, because the phosphate that is attached to nucleotide molecules, the phosphate that is found in the nucleotide, so that phosphate group, that is PO4 minus, it carries a negative charge, and these negative charges that will give a polyanion characteristic to nucleotide molecules. Remember that. And also the double bonds which are present in the purine and pyrimidine bases, there are these double bonds here. So these double bonds, they have the absorption capacity at 260 nanometer ultraviolet light. That means they absorb ultraviolet light at 260 nanometer, and this absorption of ultraviolet light by these bases will contribute to the mutation effect of ultraviolet light over nucleic acids. That may lead to certain, it may lead to a mutation which might lead to cancerous condition later on. Now, synthetic nucleotide analogues of purines and pyrimidines have been used in the clinical medicine. Some of the synthetic nucleotides that we know is 5-fluorouracil, 5-iodouracil, 3-deoxyuridine, 6-thioguanine and 6-mercaptopurine, 5-or-6-azajuridine or 5-or-6-azacitidine and 8-azaguanine. So all these are used in treatment of different types of cancer. And also allopurinol, which will inhibit the xanthine oxidase enzyme, and it is inhibiting de novo synthesis of purines. So thereby it can control uric acid levels in the body. So allopurinol is an inhibitor of xanthine oxidase enzyme directly and de novo biosynthesis of purines indirectly. Cytarabine, which is a chemotherapy drug, and also azathioprine, which is metabolized to 6-mercaptopurine. So they are used in suppression of immune rejection during organ transplantation. So those are some of the synthetic nucleotides that can be used. Like adenosine monophosphate derivatives, we have 𝐿-adenosyl-methionine, that is SAM, which can be used as a methyl group donor. SAM is one of the methyl group donor which is used in a variety of biological reactions in our body. And then we have something called as phosphoadenosine phosphosulfate, that is PAPS, PAPS. PAPS, that is phosphoadenosine phosphosulfate, is an active sulfate donor in our cellular reaction. So this is an active sulfate donor, whereas SAM is a methyl group donor. Methyl group donor is SAM, and PAPS is sulfate donor in biological reaction. And cyclic AMP, it's a second messenger, so it's a nucleotide derivative here. And nicotinamide adenine dinucleotide, flavin adenine dinucleotide, these are the nucleotide derivatives there, which are acting as a coenzyme. So coenzyme A itself is a nucleotide derivative, so which is participating in a variety of biological reactions in our body. Coming to the nucleic acids like DNA and RNA, as I have told you, these are polymers of nucleotides. That means one nucleotide joined with another nucleotide to make that polynucleotide linkase. And the link that is connecting these nucleotides together to make a polymer, to make deoxyribonucleic acid and ribonucleic acid, and that is phosphodiesterase linkase. And this phosphodiesterase linkase, it links two nucleotides together. Okay, so what kind of phosphodiesterase linkase we are looking for here? It is the 3', 5' phosphodiester bond, which will make a backbone for a nucleic acid, that is DNA and RNA. So because of this 3', 5' phosphodiester bond formation, so you are going to see two ends in a nucleic acid. One is the 5' end, other is the 3' end. So that is shown in this particular figure here, as you see. So 5' carbon here, this is the 5' carbon, and phosphate is attached, that's a 5' phosphoryl end. And then the 3' carbon of this ribose sugar, which has got hydroxyl group, and that hydroxyl group is attached with the phosphate that is present in the incoming nucleotide. So that makes a connection between 3' hydroxyl group and a 5' phosphate, making 3', 5' phosphodiester bond. This is the linkase that connects two nucleotides together, and this sugar, ribose sugar, having a 3' hydroxyl groups here. So this is a 3' hydroxyl end and 5' phosphoryl end, and that's where it is shown as 3' to 5' end here. So 5' is phosphoryl end and 3' is a hydroxyl group there. So each nucleotide, example here, adenine, thymine, guanine, cytosine, and then adenine, they are connected with one another by 3', 5' phosphodiester bond. So this is all about nucleotide chemistry, especially about bases, nucleosides and nucleotides. Thank you.